YouTube.
So can I share my screen?
Sure, sure.
Okay. Yes.
So
I don't need...
let's wait 1 second for the setup. Okay, now we are on YouTube now. So let me start with a brief introduction about Professor Shujin Li. So Professor Shujin Li received his PhD degree from University of Science and Technology of China in 2009. So after that, he spent multiple years at Brown University. The first as a postdoc scholar, and then later he was promoted to a research associate professor before he joined Zhejiang University. So now he's a professor at the Zhejiang University in the Department of Mechanical Engineering. And his research focused on the area of micro and nanofluids, cell mechanics, and blood modeling, and related topics. So Shujin, please start.
Okay, thanks for the introduction, Jen. This is Shujin from Zhejiang University. And today, my talk is on the modeling of flow dynamics of red blood cells in health and disease. And here's the outline of my talk. In the beginning, I will give you a brief introduction on blood flow and its main component, red blood cells. Then I will show you some simulation results on the flow dynamics of red blood cells in health and disease. After that, I will introduce some modeling work on the adhesion dynamics of red blood cells in sickle cell disease. Next, I will show you some recent simulation study on the viscoelastic property of red blood cells in cell aging and the mechanical fatigue process. Finally, is a brief conclusion. As we know, the blood flow through blood vessel is of viscous nature. And this viscous nature results in Stokes flow near the wall and the positive flow away from the walls. And the inertial lifting force causes the red blood cell inside a larger blood vessel to deform and finally move towards the channel center, like this way. And this leads to a cell-free layer near blood vessel wall and the myelination of white blood cell and platelet. Here, in this figure E, we show the white blood cell and the platelet in white and yellow color. But in a narrow blood vessel with its size smaller than cell diameter, the cell wall interaction would compress the red blood cells. And this leads to gas exchange with the surrounding tissue. And also we know the blood comprises blood cells suspended in blood plasma. And the blood cells include red blood cell, white blood cell, and a platelet. And a normal red blood cell has a biconcave shape. So it looks like a donut without a hole in the center, like this shape. And its diameter is around 8 micrometer, and its thickness is about 2 micrometers. And the cell membrane consists of 2 components. One is called lipid bilayer that contributes to the bending resistance to the cell membrane. And the other component is cytoskeleton or spectrum filament. And the spectrum filament mainly contributes to the shear resistance to the cell membrane. And in the healthy condition, these 2 components, the lipid bilayer and the spectrum filament, are connected by the transmembrane proteins. like the band 3. And the red blood cells circulate the human body, delivering oxygen from lungs to tissues during their normal lifespan of about 4 months. And the deformability of the red blood cells, or the ability of the red blood cell to deform its shape and apply the stress, play an important role in the main function of the red blood cells. In healthy condition, a normal red blood cell is quite elastic, so it can bend and flow freely without any damage when passing through narrow blood vessels. And we can tell from this, these 3 experimental images. We can see that the red blood cell has a large deformation as it squeezes through the narrow channel, and the cell shape changes from its normal biconcave shape to an elongated shape during the transversal process, from like this, the biconcave shape, to an elongated shape during the transversal process. And after the red blood cell move out from the exit, you can see the red blood cell recover its original biconcave shape here. Yes. However, in blood disorders such as sickle cell anemia and malaria, this general feature of the red blood cells may be changed. For example, in sickle cell anemia, the polymerization of abnormal hemoglobin inside the red blood cells causes the membrane stiffening and change cell shape. And this rigid sickle cell can block blood flow and lead to vaso-occlusion in blood circulation, as shown in this figure. So, one question is that, what's the role of the cell mechanical property in influencing blood disease? And one more important question is that, what's the role of this membrane defect in disease in influencing cell mechanical property and its flow behavior in the microcirculation? It's not clear by far. So here, we address this question through the multiscale simulations. And these multiscale simulations are guided and validated by company experiments. And in the multiscale modeling, we employ a mesoscopic particle-based method we call dissipative particle dynamics, or just called DBD. Actually, the DBD is a mesoscopic method used to simulate complex fluid behavior. And in DBD simulations, each DBD particle represents a cluster of atoms or molecules. And the DBD particle interacts through a simple pairwise potential. And also, in DBD simulations, we calculate the position and velocity of interacting particles over time, and account for the interaction through 3 pairwise force, including the conservative, random, and dissipative force of fluids, plus dissipative membrane forces. And using the DBD method, our collaborator developed a multiscale red blood cell model. And in this model, the membrane of the red blood cell is modeled by a 2D triangular network with several hundred to several thousand vortices. So we call it multiscale RBC model. And each vortex is a coarse-grained DBD particle. Like in this movie, we show the black dot. And here is a vortex, is a DBD particle. And these DBD particles are connected by viscoelastic bonds, like this, the black lines is a viscoelastic spring, viscoelastic bonds. And in the red blood cell model, we consider the bending resistance of the lipid bilayer. Use this equation. And the shear resistance of the cytoskeleton using this equation. Also, the surface area and the volume of the red blood cell are constant. So in the DBD simulations, the red blood cell can maintain its biconcave shape under normal conditions, just showing in these movies. You can see the red blood cell can maintain its biconcave shape under normal conditions. And using this DBD-based RBC model, you can simulate the flow dynamics of healthy and diseased red blood cells. To validate the cell model, people can simulate the cell flow behavior in nanochannels. Here, the left figure shows the experimental result, and the right figure shows the simulation result. We can see, similar to the experimental study, the simulation also shows the cell shape changes from its undeformable shape to deformable one when the red blood cell move into the nanochannel, right? And after the red blood cell exits from the channel, it can get back to its biconcave shape, as shown here. And we can also analyze the pressure velocity relationship for cell flow across channels. See this figure, see the right figure here. We can find that the simulation results are consistent with the experimental measurements. They show the similar trends. In the above discussion, we show the normal red blood cell can flow freely in blood vessels. And these normal red blood cells contains normal hemoglobin, like in this red dot, inside the red blood cell membrane. Here is normal hemoglobin. But under certain conditions, like sickle cell disease, the normal hemoglobin become abnormal. And this abnormal hemoglobin can polymerize into long fibers inside the cell membrane. And these long fibers distort the cell membrane and cause blood cells lose their normal shape and become sickle shape like this. It's a sickle cell. And these sickle cells are quite stiff and sticky, so they are unable to deform when they pass through narrow blood vessels, but block blood flow and lead to vaso-occlusions, as shown in this figure. So the patient can suffer a range of conditions like acute anemia, tissue and organ damage, and terrible pain and even strokes. But people don't know when the vaso-occlusion is going to happen or how to predict it's coming or not. So it's still difficult for patients to have effective care. And to analyze the biophysical alteration in sickle cell and transient hypoxia, our collaborator at MIT developed this tiny device showing here. And this device can control the oxygen level. Yeah, here shows the oxygen level. And using this device, they study the cell sickling and unsickling process. and identify cell sickling from the microscopic image shown in the middle of this figure. And the yellow arrows indicate the sickled red blood cells, like this classical sickle shapes and these granular sickle shapes. Also, all of these cells are sickled cells. Also, they designed the microfluidic channel with capillary-inspired structure for single-cell rheology study. You can see from the right figure, there are many microgates in the microchannels, and each microgate has a width of about 4 micrometers and depth only 5 micrometers. So they are chosen to mimic the size of the small capillary in human body. It's about 5 micrometer in size, right? And through this device, they can measure how long it takes blood cells to become stiff, making them more likely to get trapped in blood vessels. Here, in this figure, we show the time sequence in this direction, T. The time sequence of red blood cell flowing through the microgate. And here, the flow direction is from right to left. And the yellow arrows indicate the sickled cell that are unable to deform and pass through the microgate, thereby blocking blood flow here, right? You can see from up to down. And the normal condition, the sickle cells are deformable, so all of them can pass through the tiny microgate. So there's no blockage as a microgate. When the oxygen level is removed, we can see some cell sickle and get stuck as a microgate, from just like this one and these 3 ones. But the healthy ones can still go through the microgate, like this one can go through. And when the oxygen level is reduced further, you can see more sickle cell gets trapped as a microgate, right? But once the oxygen level is go back to the normal range, the unsickling process occurs and the cells flow in normal conditions. You can see finally there is no cell trapped as a microgate. All of the cell can get back to its normal condition and pass through the microgate again. Yeah, here's the observations. And here is a video showing the observations. You can see in a normal condition, the sickle cells are deformable, so they can pass through the tiny microgate like this, right? And when the oxygen is removed, some cells get stuck like this one as a microgate, but other cells can go through the microgate. You can see more cell gets trapped at the microgate, like this one and also this one. And once oxygen is reintroduced, the unsickling process occurs and cell can flow in normal conditions, right? And this trapped cell can pass through the microgate once again. Yeah. Here, we try to model the cell sickling and unsickling process and study the sickle cell behavior in microchannels. So here, we develop a kinetic model to track the dynamic process of cell sickling and unsickling process and transit hypoxia. And we introduce the oxygen level into this equation. See, we include the oxygen pressure, oxygen level into this equation. And through this way, we can control the cell shape and the cell membrane properties. And when the oxygen level is decreased, we find that the cell deform its shape from normal biconcave shape to different sickle ones, like the granular shape, sickle shape, and also the elongated shape. So use this cell kinetic model, we can model the cell sickling and unsickling process. And here in this figure, we show the cell sickling and unsickling process and transient hypoxia conditions. You can see that there is a shape transition from the biconcave shape to sickle shape when the oxygen level is reduced. In the beginning, the oxygen level is normal, so we get a biconcave shape for the red blood cells. And when the oxygen level is reduced, the cell sickling occurs. And at the lowest oxygen level, we get a classical sickle cell, like here. And after that, the oxygen is reintroduced. So the cell unsickling process occurs, and the cell gets back to its original shape. Yeah. So we can model the cell sickling and the sickling process through this model. Yeah. And including all of the data, we set up the simulations. And here is a video showing the cell flowing behavior in microchannel. You can see that for individual sickle red blood cells, they may have different dynamic behavior and transient hypoxia conditions. Here, yes, let's go at a normal oxygen level, just like here, you can see all of the red blood cells deformed and pass through the microgate. There's no problem. And when the oxygen level is reduced, some red blood cells get trapped as a microgate, right? And after the oxygen level is restored, the rigid red blood cell can pass through the microgate once again. Yeah, this here is the oxygen level. When it's restored, this sickle cell, this trapped cell can pass through the microgate once again. And here I put the experimental results and the simulation results together. And in this figure, we show the time sequence of sickle cells flowing through the microgate and transient hypoxia condition. Here, the red blood cell flows from right to left. So the flow direction is from right to left. And the yellow arrows indicate the trapped sickle cells as a microgate. And here, the time development. At the normal oxygen level, we find that all of the red blood cells are easily deformed and pass through the microgate, both from the experiment and the simulation. You can see all of the cells can pass through the microgate. And following a reduction in oxygen level, some red blood cells become sickled and get trapped as a microgate. Just like this one, they get trapped as the microgate. And also this one from the simulation, we find 2 red blood cells get trapped as a microgate. But other red blood cells can still deformed and squeeze through the microgate, like this one. They can still pass through the microgate, and also this one. There's no problem for it to pass through the microgate. And when the hypoxia continues for a longer time, we find that more red blood cells become sickled and blocked as a microgate. Like in this image, you can see there are a cluster of red blood cells get trapped at this microgate. And also from our simulations, we get some similar results. That 2 red blood cells get trapped at this microgate, and other 2 red blood cells get stuck there. And after the oxygen level are restored, these rigid blood cells once again traverse the microgate. You can see that less red blood cells get trapped there. Finally, all of the red blood cells can pass through the microgate. Yes. And also through this study, we find for individual sickle cells, they may have different behaviors. For the dense and rigid sickle red blood cells, they tend to get stuck and clog the blood flow. Like this one, the yellow arrows in the shows, yeah, this sickle red blood cells get trapped as a microgate and clog the blood flow. But for the light and deformable sickle red blood cells, or healthy red blood cells, they can choose a smart pathway when they have support some trapped cells ahead of them. They just go right through the obstacles. Just here, we show the trajectory of deformable sickle cells. You can see that these cells can go right through the obstacles. They can still pass through the blockage. Yes. And from the above this discussion, we know that the cell sickling can cause vaso-occlusion in microcapillary. So here in the microcapillary, the cell sickling can cause vaso-occlusion. But in a large vessel, like postcapillary or venule, the size of this blood vessel is much larger than that of sickle cells, right? For example, this diameter is larger than the sickle cell size. So the cell sickling is necessary but not sufficient to trigger blood occlusion. Instead, the adhesion between the sickle red blood cells and vascular endothelium play a key role in triggering vaso-occlusion. So the cell adhesion is important. Finally, it leads to vaso-occlusion in the postcapillary venules. For example, in mice, the adhesion and aggregates among different cells, including the red blood cells shown in green color in this domain, could lead to blood occlusion in larger blood vessels, right? You can see this domain, there's no blood flow. The blood flow stops here in this domain. So the cell adhesion interaction is important for triggering the vaso-occlusion in larger blood vessels. But it's hard to quantify the cell adhesion dynamics and the adhesion strength inside the body, right? So here we quantify the adhesion of sickle cells by combining experimental and simulation studies. And in the simulation, we use a kinetic adhesion model. And this kinetic model is a stochastic bond formation and a dissociation model. You can see from the left figure, on the cell membrane, there are many blue dots. And these blue dots represent the receptors. And on the blood vessel walls, there are many yellow dots. And these yellow dots represent the ligand coated on the blood vessel walls. And in the simulations, the blue dots and the yellow dots can form dynamic bonds depending on the formation rate and the dissociation rate. And the formation rate and the dissociation rate are shown in these two equations. And if a bond is formed, the force between the receptor and the ligand is adopted in this equation, like a hooking springs. And we employ the adhesion model to quantify the adhesion property of sickle red blood cells. And here there is a figure showing the result. And here the black arrow denotes the flow directions is also from right to left. And the cells inside the dotted circles are adherent cells. As you see here, there are many cells adhere to the blood vessel walls. And from the measurement, we have two main findings. First, we find the hypoxia can enhance the adhesion of sickle red blood cells. And from this table, from the table on the right, you can see more cells adherent to the wall surface and hypoxia conditions, like in these numbers. And these numbers are much higher than those of normal conditions, right? The second finding is that the age of red blood cells also plays an important role in regulating their adhesion dynamics. A deformable cell, like the round ones, is the deformable sickle cells, can form multiple side adhesion. But for a rigid cell, like these sickled ones, usually form single side adhesion. And here we show the detailed adhesion process. And for deformable sickle red blood cells, in the beginning, it has only one adhesion site in here, in the white dotted circles. Over time, we find that some additional adhesion sites are formed. For example, at 2 minutes, there are 3 adhesion sites for these deformable sickle red blood cells. And our simulation reproduces this process. You can see in the very beginning, there are only 1 adhesion site for these sickle cells. Then more adhesion sites formed. Finally, at least 5 adhesion sites for these deformable sickle red blood cells. And in the right figure, we show the bond counts during the dynamic bond formation and the dissociation process. You can see that the adhesion site increase, then approach a constant with time, right? This is for the deformable sickle red blood cells. But for rigid sickle red blood cells, in the very beginning, there's only 1 adhesion site. But with time, there's still only 1 adhesion site. We find that once the cell adheres to the cell surface, it's flipped around the adhesion site, just turn around, and to align with the flow direction, but no new adhesion site forms, right? So for the rigid sickle red blood cell, it's always single site adhesion, and our simulation can reproduce this process. Even there are some temporary adhesion site forms, but it's not steady. Finally, still only 1 adhesion site for the rigid sickle red blood cells. Then, when quantify the cell adhesion strength, here in the left figure, we show the sketch of the simulation setup. There's a red blood cell is adhered to on the wall surface. And here, the green color represents the ligand coated on the wall surface. Through this simulation setup, we quantify the cell adhesion strength by increasing the shear stress until the onset of cell detachment from the wall surface. Finally, you can see the cell detached from the wall surface. Then we can get the shear stress threshold and estimate the cell adhesion strength. And here's the video showing the cell detachment process. We find that the adherent cell is deformed and shear, right? Then increase the shear stress. The weaker adhesion site is ruptured first, like this, the yellow dotted lines. This is weak adhesion site, so it's ruptured first. And then the cell holds onto the stronger adhesion site and shear. The red blood cell with single site adhesion obtain an elongated shape like a teardrop, right? Finally, the cell detach from the wall surface when the applied shear stress is above the threshold, right? Finally, it's detached from the wall surface. And the simulation shows a similar phenomena. For example, in the very beginning, there are two adhesion sites here. The weaker adhesion site break first, so the cell hold become elongated shape. Finally, the cell detach from the wall surface when we use a higher shear stress. And through this measurement, we find that the shear stress threshold for the deformable sickle cells is about 4 to 5 times higher than that of rigid sickle cells. For example, it's about 4 times higher than those of rigid sickle cells. Hence, we conclude the deformable sickle cells have greater adhesion strength compared to those of rigid cells. And this confirms a previous hypothesis based on the DBD simulation study. And in this DBD simulation study, our former collaborator, Fanli and George Kalianakis, simulated the adhesive dynamics of sickle red blood cell in shear flow. For example, in this figure, the blue cells are deformable sickle cells, and the red cells are the rigid sickle cells. You can see from this video, the deformable sickle cells adhere to the blood vessel wall first. Just adhere to the blood vessel wall first. And this can trap the rigid sickle cells and lead to blood flow occlusion. Hence, they stated that the interplay of deformable and rigid sickle cells can trigger full blood occlusions. Our results confirm their statement. Yeah. The last part is about the modeling of red blood cell viscoelastic property in cell natural aging and mechanical fatigue. As we know, in cell natural aging mechanical fatigue process, the cell deformability and the membrane viscosity changes. For example, there is cell membrane remodeling in cell aging process. And this would cause the rupture of spectrum filament, like this one, the spectrum filament rupture. And small pores merge into larger pores. For example, in the left figure, you can see many pores there. But in the right figure than some larger pores. And this leads to increased cell membrane stiffness. Also, the cell membrane becomes stiffer in mechanical fatigue process. For example, in a recent study, Chan et al. Studied the mechanical fatigue of human red blood cells. They show that the red blood cell gets stiffer. You can from the right figure shows the shear modulus increase with the number of fatigue loading cycles, and also the membrane viscosity also increase. So the red blood cell membrane become more viscous with the number of fatigue loading cycles. Here, we develop a multiple-step framework for cell modeling. First, we collect the experimental information on the structure of the cell membrane. Like here, gets this experimental structure information of the cell membrane. And this can guide us to set up the cell membrane model. Like here, consequent cell membrane composite model. And using this model, we can compute the shear modulus and the membrane viscosity of the cell membrane. Next, when we get this mechanical property of the cell membrane, we can pass them to the DBD-based whole cell model and quantify the alteration of cell behavior in cell natural aging and mechanical fatigue. Here's the fourth step. We get the nanostructure of spectrum network taken by AFM test. Then we employ an imaging skeletonization method to convert the AFM image into spectrum structure network in the middle. Next, when we get the spectrum structure network, we can set up the consequent cell membrane model. Yes. Then we use this structure information to guide us simulation setup. We can build a consequent component cell membrane. And using this cell membrane model, we can compute the viscoelastic property of the cell membrane. For example, to compute the shear modulus of the cell membrane, we let the cell membrane subject to shear at a fixed shear strain rate. And the shear modulus can be estimated from the shear stress and shear strain curves. We can get the shear modulus. And the membrane viscosity can be computed through the green combo formula in these two equations. And here, in this figure, we show the trends of shear modulus and membrane viscosity with the cell aging. It shows that the shear modulus increased from 10 to 16 with cell aging, and the membrane viscosity increased from about 150 to 400 with cell aging. So we can confirm that the cell membrane become more stiffer and more viscous with cell aging. With this key parameter as model input, we can employ the DBD-based RBC model to quantify the viscoelastic response of red blood cells. For example, we can let the cell subject to stretching deformation, like the optical tweezer experiment. We find that the red blood cell deform its shape and stretching from the left to right. And when the stretching force is released, we find that the cell can get back to its initial shape. Hence, the cell viscoelastic response can be obtained in the shape relaxation of the red blood cell membrane. Then we compute the relaxation time and compare with cell fatigue experimental measurement. You can see here they show some similar trends. Right? Finally, we probe the dynamic response of the red blood cells and different loading modes using twisting torque cytometry. And in the model setup, we just put a solid microbead, in the yellow microbead, attached on the top of the cell surface. And for the microbead, is subject to oscillating field. So when we applied an oscillating torque to the microbead, the displacement of the cell membrane shows the same frequency response, but shaped with a delay phase. Then we can compute the storage and loss moduli through these two equations. Right? We find that for the storage modulus, they show a flat plateau at a low frequency. This shows that the cell membrane is nearly elastic in this region. But for the older red blood cells, it have higher value of storage modulus, suggesting a great shear stiffness of cell membrane. Right. Also, the behavior of the loss modulus can be fitted by a power law, like so here. And for the old red blood cell, we obtained higher value of loss modulus compared to the younger red blood cells, which again confirm that the older red blood cells have higher value of membrane stiffness and viscosity. Okay, it's bring my conclusions. First, by combining dynamic experimental measurements with multiscale simulations, we study the flow dynamics of healthy and diseased red blood cells in blood vessels that range from narrow capillary to venous. Also, we develop a multiple-step framework for cell modeling. So this multiple-step framework can be used to predict the viscoelastic property of red blood cell in cell aging and mechanical quality. Also, this framework can be extended to quantify the dynamic mechanical behavior of biological materials. Okay, thank you for your attention. Yeah, I'll stop here.
Thanks, Xuejin. It's a very comprehensive introduction about the blood modeling and also the cell mechanics modeling. So any question from our audience, you can unmute yourself to ask a question directly to Professor Xuejin, okay? So you can, if you have a question, you can unmute yourself.
So hi, Xuejin, I have one question.
Go ahead.
So for in the last part, when I go from the image of the red blood cell membrane to the modeling structure, yes, right, yeah, this slide, the next one.
Yes.
So why so for the right top corner, the image here, it seems to me you, let's say, increase the map or increase the resolution of the bonds in some triangles. but instead of decrease the area of each triangle?
Yes, actually we try to decrease the triangle area, but we couldn't see bigger difference between them. So we changed our idea and just to dense some local triangular network.
Then how do you determine which two nodes should I add a new bond?
Just randomly choose based on the spectrum network connectivity. We just increase some of the vortices.
Okay, I see. Thank you.
Yeah.
Oh, hi, Xuejin. This is Zhao Yan from University of Buffalo. So in fact, it's you are giving a very nice talk, and I do have two questions about your work. So the first one, in fact, is about the RBC model. So I remember that you have a slide shows the RBC model. Would you mind to go back to that one?
Sure. This slide.
Yes, that one.
Yes.
Okay. From here, so here you mentioned that the surface area and also the volume controls the last one, the potential of the last formula, right? So yes. I'm just wondering. So say if you want to make a smaller triangle by using more like a fine, like a mesh to represent the RBC structure. So is there any other parameter we need to pay extra attention for that part?
That's a good question. I think if you want to use a smaller triangular to represent the cell model, I think you change this parameter.
Okay, so this A0 is the surface area.
A0 is just like is for the single triangular angle. Here it shows the area for each one, and in the last two terms, in the middle terms, it shows the total area for the cell membrane.
Then does this model require very uniform like meshing network for this model, RBC model? Say if I have very non-uniform, like some kind of very bad shape of the triangle in this.
Actually, it's a multiscale read blood cell model, so you can change the triangular network. Just you can use over dense network. You just like you dilute.
Yeah.
But you change some parameters. Just
like
right. Zero.
Yeah. But I'm wondering, so for this, for example, I would like to have some certain elements to represent the network, but my triangles in this network are not very uniform. They are not quite similar with each other. So will that influence the stable structure?
I'm not sure, but if you change just like some triangular area, there may be for some case they may be unstable, but for most of the simulation, they're okay, they're fine. You can use different triangular area for a different triangle.
I see. So to be specific, for this model that you are showing here, so how many like particles and elements that you use in this model?
In this video, we just use I think 500 DBD particles, just 500 vortices on the cell membrane. It's a close-range cell model, but we also have a finer resolution of the cell model. So like, for example, we can use more than 20,000 particles for the RBC.
Okay. Yeah. So my second question, in fact, is about the coupling of the oxygen level with your DPD model. So you have a kinetic model, like you showed, the oxygen concentration or something. So
Yeah.
I'm wondering, what is the mu here, and how you integrate this mu to the forcing term in your DPD model?
Yeah. Here the mu is the shear modulus of the cell membrane. So for the sickle red blood cell, this mu is much higher than the normal ones. So, but mu zero is just like for the normal red blood cell. You can see through this equation, if we decrease the oxygen level, this term will increase. So finally, we will increase this mu. This mu, if this value is higher, that means that the cell becomes deeper.
Becomes deeper. Okay. So then this mu factor, will the mu factor appear in the forcing term of your DPD equation? Or
in the cell model, because we use just like the shear modulus for the cell membrane is included in the cell model.
Okay, I see. And by the way, how do you define the sick, like sick RBC? I mean, a rigid Sickle RBC and the deformable, like Sickle RBC. It's just purely depends on the shape, or
not only depend on the shape, but also depend on the mechanical property of the cell membrane. For example, for the deformable sickle cell, we just use a smaller shear modulus. But for the rigid sickle cell, this value is quite high.
Oh, I see, I see. Okay, thank you very much.
Welcome. Any other questions?
Any other questions from our audience? Okay, Shujin, I want to ask you a question about the cell adhesion dynamics. You mentioned for the rigid cell, usually the binding or the adhesion usually is a single site. Yes. And if you have soft cells, usually, for example, the healthy one is pretty soft and deformable. You have multiple site adhesion. So this means that the healthy or the soft cells have larger adhesion force attached to the vessel wall. Then actually for the sickle cell it's easier to block the flow, right? But the modeling part or for the adhesion part, the sickle cell actually have weaker adhesion to the vessel wall. So what's the main reason for the sickle blood? The blood with sickle cells have easier, and it becomes easier to block the entire blood flow in the vessels.
Yeah, actually, I think in Juan's simulation study, they show this for the deformable sickle cells, they are more adherent to the blood vessel wall. So it's easy for this kind of cell to attach on the blood vessel first. Then it will cause the effective size of the blood vessel become smaller, and this can trap the rigid sickle cells. And this will lead to the blood occlusion finally.
Okay, Okay, so it means that the vaso-occlusion phenomenon is actually not because of the sickle cell attached to the wall. It's because the soft deformable cell attached to the wall, and then the rigid cell block the other region, right?
Yeah, because the blood vessel diameter is larger than the sickle cell size, so it's hard for the sickle cell block at the larger blood vessels. So in the first step, the deformable cell needed to attach to the cell the vessel wall, and just reduce the effective size of the blood vessel. And in this way, they can trap the rigid cell. And also for the healthy red blood cell is hard to form adhesion. Only for the deformable sickle cells, they are adherent to blood vessel walls. For the healthy cells, it's hard. So there's no adhesion between the healthy red blood cell and the blood vessel walls.
So for the vaso-occlusion phenomenon, that's the bigger cells. For example, white cells is also playing a role in the dynamics, or
yes, that's true. Because the white blood cell also can adhere to the blood vessel. So if there's some white blood cell, for example, in this video, you can see that here the neutrophils, just like the white blood cells, you can see they adhere to the blood vessels, blood vessel walls, and form aggregates, and this contribute to the blood occlusion as these larger blood vessels.
Okay. So the white cell also play an important role in this dynamics process, right?
Yes, just like in this figure show here, there are some. I think this kind of cell is white blood cells, and this is the sickle cells. Because the white blood cell has a bigger size, right?
Yes.
Yeah, you can see from here, the white blood cell is there.
Okay, thank you, Xuejing. So any more question from our audience? Okay, if not, then we will stop here today. So if you have additional question, you can contact Professor Xuejing directly. So the contact information actually we can also post on our website. You can link Professor Ni's group website from our CFSM website. Okay, thank you all for attending the seminar. Bye-bye.